Good afternoon. My name is Carolyn Becker, and I'm a senior team lead in the Translational Medicine Department at QPS located in Newark, Delaware. I'm excited to share our recent experience with the SARS-CoV-2 serology assay. The SARS-CoV-2 virus is part of the coronavirus family, which causes the severe acute respiratory condition referred to as COVID-19. There are 6 additional coronaviruses capable of infecting humans currently. It is a positive sense single-stranded RNA virus and contains 4 structural proteins: the spike protein, the envelope protein, the membrane protein, and the nucleocapsid protein. SARS-CoV-2 gains entry into a host cell via the ACE2 receptor found on human cells. Early in the onset of the COVID-19 pandemic, it was widely acknowledged that a gap existed between the molecular tests available and the need for data by the public health authorities to make informed recommendations. 6 months into the pandemic, we've made significant progress to meet demand for accurate PCR and serology testing services, as well as advancement towards effective drug therapies and vaccines. So what is serology? It's the study of sera, usually to identify antibodies made in response to an infection. The IgG antibodies can be reliably detected around 14 to 21 days post-infection. While serology testing is not effective for identifying people with active infection, it has a wide range of applications. The FDA issued the current EUA guidance on May 4th, 2020, and superseding a similar policy released by the FDA on March 16th. It recommends the following validation tests: cross-reactivity or analytical specificity. This is to ensure that the assay does not cross-react with the presence of antibodies against other common viruses. Class specificity, to ensure that the class of antibody detected is recorded as labeled. And clinical agreement, which assesses sensitivity and specificity of the assay. Sensitivity, or percent positive agreement, estimates the percent of PCR confirmed positive samples which are screened positive in the tests. And it's expected to exceed 90%. Negative percent agreement estimates the true negatives, which are screened negative, and it's expected to be greater than 95%. The FDA had summarized the performance of the existing serology tests, which have received an EUA, and you can find that located on the FDA website. Generally, sensitivity and specificity have been recorded across a wide range for the different tests, but there are multiple tests now reporting specificity greater than 100%, and most have specificity between 95 and 99%. Individuals in the ADA bioanalysis field have spent years discussing and refining the optimal approach for validating ADA methodology with the finalized FDA guidance released in February of 2019. It is relevant to apply the industry experience with ADA in order to design a specific serology assay. During the development phase, there are many similarities between the ADA method and a serology assay, identifying a well-characterized, purified capture antigen and selective detection reagents, as well as use of effective positive and negative controls to assess assay performance. Additionally, it's important to evaluate and identify the best buffer composition in order to minimize background without diminishing specific signal. And finally, optimizing reagent working concentrations, including the use of a minimum required dilution to mitigate nonspecific binding in the assay. Progressing to the validation phase, the approach to characterize and describe the performance of the serology assay diverges from our ADA experience. Performance of a serology assay is primarily described as sensitivity and specificity. Although these are familiar terminology for most accustomed to ADA assays, they actually have a contrasting meaning for serology assays. Sensitivity is essentially the true positive rate, while specificity is the true negative rate. The established sensitivity and specificity of a serology assay are only estimates, and they're limited by the samples that are used to validate or characterize the test. Therefore, it's recommended to report the 95% confidence interval for these performance measurements based on the number of samples that are evaluated. The more samples of known positive or negative origin that are available for validation, the smaller the confidence interval becomes, and it's Because of the variation among individuals between the onset of infection and seroconversion, a well-characterized positive sample demographic can be crucial in understanding an assay limitation. We sourced a variety of matrix from commercial vendors to support the validation. We received 165 lots of normal negative matrix that were collected prior to December of 2019. So they shouldn't have any exposure to the SARS-CoV-2 virus. The positive matrix that we received included the date of serum collection and either the date of the first SARS-CoV-2 PCR test or the date of COVID-19 diagnosis. These dates are used to calculate the days elapsed, which helps to estimate the period of time that a patient may be generating antibodies. In some cases, additional information was received on severity of the disease or date of onset of symptoms as well. To better understand how the assay performs in the presence of other viral infections, we acquired matrix collected during peak cold season, supported by the CDC published surveillance data demonstrating a 2 to 6% incidence of common coronaviruses over this period. Additionally, we sourced 5 lots each that had known reactivity to HPV, HCV, HIV, ANA, and influenza B. For the QPS approach to the serology assay, we selected the spike protein to evaluate the capture antigen. We evaluated various recombinant proteins, the S1 and S2, which includes the extracellular domain, the S1 protein alone, as well as the S1 receptor binding domain. The receptor binding domain antigen was selected as it's the most unique among the other coronaviruses and contains the region needed to bind the ACE2 proteins on the cell surface. Antibodies targeting the RBD portion of the spike protein are most likely to have a neutralizing effect on the virus, making it a specific and highly relevant antigen. We're utilizing a standard immunoassay format with HRP detection, TMB substrate, and a colorimetric readout. Due to the differences that we observed in background signal and variation among negative matrix, we intend to calculate separate cut points for different detection reagents like anti-IgG or anti-IgM, as well as different matrices like serum or plasma. Much of the focus on ADA method recommendations surround the setting of an assay cut point to distinguish a positive result from a negative result. An ADA assay, which is intended to support safety and efficacy during clinical trials, should prioritize minimizing false negatives in the assay. It's recommended to use a multi-tiered approach with a screening assay that's designed to identify a false positive rate of approximately 5%, followed by a confirmatory assay which confirms reactivity to the therapeutic protein, targeting a false positive rate of 1%. It's also recommended to perform outlier analysis on the drug-naive data set used to calculate the assay cut points to reduce variation and set a more conservative limit. In contrast, a serology test should prioritize minimizing false positives in the assay, but balance sensitivity. We recommend evaluating the known negatives and the known positive data set in combination. Consider including all of the negative sample signals in the data set when calculating the cut point and targeting the false positive rate to be less than 2% to prioritize specificity in the assay. A receiver operator characteristic curve can be a useful tool. It will graph the sensitivity against the inverse of the specificity using all instances of the positive-negative separation cut point in order to target the optimal balance between sensitivity and specificity. Where the cut point is set will impact the estimated sensitivity and specificity. There is limited information published about setting cut points for serology assays, but based on a literature review, many opt for a fixed cut point based on either 3SD or 6XD of the negative data set. Here we show the effect of the outlier removal in different statistical approaches. In most cases, the sensitivity is not significantly different when shifting the cut point, but specificity, or the false positives, will change dramatically. So I have highlighted here the 99.9% non-parametric cut point, and it shows that we have sensitivity at 92%, specificity at 100%, and cross-reactivity at 95% using this cut point. You can see if you remove one round of outliers, which would be identified in the histogram to the right, that we see a significant decline in the specificity and cross-reactivity negativity, whereas the sensitivity or the positive samples really aren't affected too much. Part of the reason for the minimal shift in the sensitivity with different cut points is that most of the positive population has clear, strong reactivity in the assay. Less overlap between the populations results in higher estimates of sensitivity and specificity. So if you look between the 0 and the 20 axis, you can see that there are a number of positive samples that are overlapping with the plot on the negative samples as well. And while they are insignificant in the total number overall, they do cause us pause as to where we should set our cut point. So by setting the cut point in at about 7, which is what was calculated using the 99.9% rate, we see a handful of false negatives out of the positive result and minimal false positives out of the negative results. In some cases, the low reactivity for confirmed positive matrix can be explained by the sampling time. In looking at the distribution of the signal-to-noise ratio of our positive data set compared to the days between the sample collection and disease confirmation, there is not a very clear correlation, but we may not have enough information about the timing or the severity of infection. When we calculate the clinical agreement after subgrouping the positive samples, we can demonstrate that the sensitivity is in fact influenced when a sample is taken. A longer period for seroconversion correlates to the increased clinical agreement of positive samples. So you can see in the first portion of the chart that samples collected 1 to 13 days after confirmation of disease gives a percent positive agreement of only 70%. As we shift to the right and see the 14 to 21 day period, we see an increase of that positive agreement, and 8 out of 9 or 88.9% of our positive samples are now positive in the assay. And finally, after a 3-week period is allowed for seroconversion, we see an increase of the percent positive agreement to 97.8% or 45 out of 46 of our samples. In conclusion, we have validated an immunoassay specific to the SARS-CoV-2 IgG antibodies. We estimate the performance of the sensitivity to be 92.4% with a confidence interval as high as 98.8%. We estimate the specificity of the assay to be 98% with a 95% confidence interval as high as 99.6%. The assay does exhibit low levels of cross-reactivity among matrix from individuals with confounding viral infections, but the cut points have been set to minimize the false positive results. We are continuing to receive additional PCR-confirmed SARS-CoV-2 positive matrix from our vendors as it becomes available, and additional matrix from patients with confounding viral infections to better characterize our assays. We plan to continue to develop and validate additional methods for IgM-specific detection, as well as for use with the human plasma samples. We are considering development of a bridging assay format, which will allow us to detect all classes of antibodies. And finally, we would also consider a tiered approach, combining either IgG and IgM detection, or developing a second tier assay using an alternate capture antigen to maximize the identification of positive samples. I would like to thank everybody at WRIB for the opportunity to take part in this session, and the scientists at QPS who've worked hard in the lab to generate this data, especially Ankita Salvi and Isabel Carlazzi. Additional appreciation to the QPS TLM department, who provided lots of support with urgent purchasing requests and constant receipt of specimens and reagent. And of course, a big thank you to the support of QPS management and marketing. Thank you.